Good afternoon, everyone. Thank you for joining this seminar. Today, I would like to share our most recent work on the analysis of recombinant adeno-associated virus, or RAV, viral vectors using LC and MS technologies. In this presentation, I'll give a brief introduction on the background and challenges in gene therapy and its analytics. Then I'll take a deeper dive in the LC-MS of RAV capsids, including the intact protein analysis by LC-MS for protein ratio and identity test, and also the peptide mapping for sequence confirmation and the analysis of PTMs, or post-translational modifications. As we know, gene therapy has been growing rapidly in recent years, and just in 2019, there were 3 approved drugs available in the market, with many more under development in the pipelines. And because of the recent advancement in gene editing technologies such as CRISPR, the gene editing process have become so much easier, and therefore a lot of efforts have been devoted into developing the gene delivery technologies, such as viral vectors. And here, to my understanding, the delivery mechanism using viral vectors is to use a modified virus to carry the therapeutic genes and then deliver into the malfunction cells and restore the cell function. So currently, there are a lot of the viral vectors being available for developing the drug product, and in which the most popular one is the adeno-associated virus, or AAVs. It is definitely the most unique one because it's considered as non-pathogenic. That means it's one of the safest viral vectors being used for gene therapy. And because safety is such a big concern for either the regulatory agencies or the drug manufacturing companies, the analytics becomes so critical for the development and manufacturing of the viral vectors, from candidate selection to gene editing, purification, formulation to clinical trials. And the stage appropriate or the fit for purpose analytical tools are highly desired for the characterization, process monitoring, stability assessment, and release testing of the gene therapy products to ensure their safety and quality. And as you can imagine, there are many challenges in developing the analytical techniques for gene therapy products. So here, using AAV as an example, we can see that the biggest challenge is that the molecular weight is so much higher compared to the conventional biologics such as monoclonal antibodies. And that means the structure is much more complex. And the AAVs have 13 common serotypes and many more under development. And it consists of the viral proteins as the capsid and also the enclosed genome. And this complex structure definitely makes the analytical development very difficult. And in the development of conventional monoclonal antibodies, the analytical methods are commonly well established, and LC-MS plays a major role. And typically, the sample supply is unlimited. But in AAV development, there are not a lot of prior knowledge in the analytics. And what's even worse is that the production sometimes can have some problems, so that the product titer is very low, and that means really limited sample amount for method development and process monitoring. So later in this presentation, I'll demonstrate how we use the advanced LC-MS technologies to overcome these challenges in the analysis of AAVs. Over the past few years, our group has been focusing on developing the workflows for AAV analysis, including the aggregation analysis using SCC and empty fold analysis using iExchange, TOF-MS, and CDMS, as well as the plasmid purity analysis using iExchange. But today, I would like to focus on just 2 areas, which are the identity and ratio tests using RPLC-MS and also the PTM analysis using peptide mapping. So using these two technologies, the structure and function relationship can be well characterized for the AAV products. So the capsid proteins can not only protect the packed genome, but can also directly impact the viral infectivity. So the ratio, identity, and purity of these viral proteins needs to be well characterized and closely monitored. And here is showing one AAV virus contains about 60 copies of proteins, including VP123 in a 1:1 to 10 ratio. Their molecular weight is in a reasonable range, like from 60 to 80 kilodalton, but the sequence homology is really high among them. And as shown here is a screenshot from Pfizer's presentation at WCVP. Compared to standard SDS page gel method, the RPLC MS method can identify the impurities in the sample and to support the process monitoring. But the resolution here kind of needs to be improved. So the method development are facing multiple challenges, including sample-related and method-related. The AAV samples are typically having a much lower concentration compared to conventional biologics such as monoclonal antibodies, and also contains surfactants in it, which can cause interference in MS detection. And the currently available RPLC methods are suffering from resolution commonly, and there are also HELIC methods available, but requiring specific sample matrix, meaning that additional sample prep is needed. The method development work was done on the benchtop LC-MS instrument called BioAccord, and starting from the golden standard method, we're using a BHC8 column with a 0.1% formic acid. We can see that there are no separation between the VP proteins, and it's also co-eluting with the detergent or surfactant. So switching the mobile phase from formic acid to difluoroacetic acid, which is another mobile phase modifier, we found that the peak shape are improved and also the resolution is increased. Other than the mobile phase, we can see that the column chemistry also has an impact on the separations. So here, evaluating a series different bonded phases including C4, C8, and C18 columns, we found that the C4 column provides the best separation for the critical pair peaks, which is like VP1 and VP2. With the optimized method, we can see that these separative peaks or VPs can be detected by MS to get their accurate mass, including VP123, and there's a fragmentation of VP3. We call it VP3 clip. And looking more closely, we can see that some of the PTMs were well separated from the native forms in MS. For example, here's the phosphorylation, and we found this PTM on both VP1 and VP2. And with the well-separated peaks, we can monitor or calculate the protein ratios by directly integrating the peak area and the optical detection. So with the UV detection, we were able to quantify the proteins by using 0.5 micrograms of sample. And then to further improve the sensitivity of this assay, we decided to switch to fluorescence detection. And here is showing the chromatograms of using 0.05 micrograms of sample. And you can see that the sensitivity is still pretty good. So the improvement in signal-to-noise ratio after calculation was about 50-fold higher compared to using UV detection. And other than the higher sensitivity we saw when transferring from UV to FLR, we also noticed that there was the change in protein ratio by directly calculating the integrated peak area. So the VP1 and VP2 ratio were kind of lower here, and we suspect that it could be due to the differences in the UV or fluorescent active amino acids in each VPs, and it can also impacted by the phosphate group in VP1 and VP2, since they can quench the fluorescence coming from the tryptophan. So here I want to show you guys a case study to demonstrate how the developed workflows can be used for the comparability studies to support the process development of AAVs. And here is a collaboration with Dr. Xiaoying Jin from Sanofi Framingham. And basically, they found that there were two batches of AAV samples, sample 1A, 1B from batch 1, and sample 2A from batch 2. And they observed the different potencies between these two batches. So we were thinking maybe we can compare the analytical profiles to see if there are any change between these two batches, including the reverse phase LC profile and protein ratio, and also the phosphorylation level of the VPs. And using the optimized method, we found that sample 1A and 1B are highly comparable. But the first thing we notice is that the sample 2A has a much lower intensity compared to sample 1A, 1B. And when overlaying the chromatograms of these three samples, we found that the relative abundance of VP2 seems to be lower in sample 2A. So we made the comparison in a more numerical way. In the bar plot of the relative abundance for each VP proteins, we can see that the sample 2A does have a lower percentage of VP2 and a higher percentage of VP3 compared to the other two samples. And from multiple measurements, as shown in the arrow bar here, we can tell that the difference we saw are really from the sample, but not from the method variation or anything. And the results were also agreed with the CESDS analysis. And on the other hand, the phosphorylation levels were also different in sample 2A. And we can see from this bar plot here that the sample 2A has a higher level of phosphorylation level in VP2 and a lower level of phosphorylation level in VP1. So currently our collaborator is looking into this to see if it has any impact in the vector functions such as the transduction efficiency. So the next part I want to talk about is peptide mapping. We know the purpose is to confirm the sequence of AAVs and also identify the PTMs. And in addition, it can also be used for disulfide mapping and HCP analysis. So on this slide is showing a paper that demonstrating the high correlation between vector activity versus one of the PTMs, which is deamidation. And this underscores the importance of performing peptide mapping for PTM analysis. And typically, the process is just to digest the proteins to peptides and followed by LC-MS analysis. And seems to be straightforward, but the established AAV digestion methods commonly using 10 to 20 micrograms of sample, which is really much smaller compared to conventional protein digestion method, but still is a manufacturing burden. So our goal is to develop a reproducible peptide mapping or digestion methods with less than 5 micrograms of sample. And through a lot of method development work and troubleshooting, we finally came up with the workflow or optimized methods for using 1.25 micrograms of AAV samples. And here, as you can see with the development methods, we were able to get a clean peptide mapping chromatograms and also high sequence coverage, which is more than 95%. And all of the N-terminal peptides were identified for VP1, 2, 3, including the really small one, the VP2, which is only less than 500 Dalton. And in the fragmentation data, we found extensive B and Y ions, meaning that we have found all three N-terminal peptides or all three proteins with really high confidence. With the developed digestion method, we were able to identify some of the PTMs. And here is just showing an example, the fragmentation spectrum for one of the deamidated peptide. And it's worth mentioning that most of the peptide mapping work were done on the benchtop Bell Accord instrument, but there are high resolution MS available certainly if you want to look at the data more in detail. With the identified PTMs, we were able to make the quantification for process or product monitoring. And here is just showing the bar plot of deamidation and oxidation relative abundances on each individual sites. And through multiple sample preparations, we found the variation in PTM levels are very small, which is within 0.4% for the standard deviation. And the deamidation oxidation levels are also seems to be at a reasonable range, and this suggests the method-induced artifacts are at a minimal level. And in addition to deamidation oxidation, we're also able to identify additional PTM such as acetylation and methylation or phosphorylation. And here lists the site information across the amino acid sequence. So to summarize, we have developed a series of LC-MS-based workflows for AAV analysis, and you can find more information in either the booth or at Waters' website. And at last, I just want to thank our colleagues at Waters as well as our collaborators at Megadalton and also the collaborators at Sanofi and BioReliance for providing the sample and serotype information. And thank you for your attention.